Okay, I'm going to go over what I've talked about before with bar plots and graphing values. I think I'm actually going to combine them into here because these ones are useful for previous ones, but the Q10 calculation is going to be a separate video because it's not for this lab report, really. Q10 calculation is going to be the slope of this relationship right here. And the way that you can calculate it from these values, because this is an exponential relationship, there is a specific value that you can actually use to convert these into a Q10 value, which is instead of it being in log e, is going to be in doubling rate per 10 degrees centigrade. So it changes, but I'm not going to go over that. I'm going to be going over how to report these values in your lab reports. So I'm going to go over some help on your lab report. This is a good cheat sheet on how to actually get ready to write your manuscript, and then how to get feedback and everything, how you should funnel your background and everything like that. We'll be going into this as we go along. I'm going to be going through the rubric and talking about individual sections, and then I'm going to have a separate little chapter on how to run your statistical tests and how to plot your values and plot your graphs, so how to actually produce these. So we start with the title page. If we go into the appendix, which I would always send to you guys, if you're going to pre-submit, be sure to pre-submit. You can just submit your lab report up to 24 hours before by emailing me, and by emailing those pre-submissions, I will grade it across this rubric, and then I can't give you the total number of points, but I can tell you how much you would earn, and that would be a guarantee as long as all of those points are contained in your actual final report. So it's a good way of understanding how this is going to be graded out, and what's going to be the most important part. So please pre-submit. You can pre-submit as many times as you want up during that week beforehand, but up to that final point, I'm going to be starting to get swamped in those. So just know that I accept pre-submissions, and you should at least get one of these back before you do a final submission. So in order to do well on these lab reports, you should follow the appendix and you should follow the rubric. So the first title page, you would include your name, the biology and your section number, the title, date performed, and the due date. And whenever you perform in a lab, be sure to include your lab partners and include my name, just so we can know which class this was in. There is no abstract, so you're not going to have to write out, well, this is not one with an abstract. You don't have to write out an entire abstract for your papers. This is a paper that's related to what we're looking at, but this is actually with an endothermic moth. So if you remember, certain moths are endothermic, while most insects are actually poikilothermic. Well, exothermic. Their body temperature is going to change their environment. Well, these ones are actually producing their own body heat, as you can see in here. So these are special exceptions to insects that are actually increasing their body heat, but because of that, they can actually plot these relationships based off of the actual by the actual metabolic rate rather than the temperature. So instead of controlling temperature, they're actually plotting temperature against this change in CO2 consumption. But so these axes are flipped from what we would normally see, but they represent that same relationship that we're actually plotting on here. And I think that this paper is really interesting, so that's why I'm including it as an example for these. So no abstract required. Title page is a separate page. So whenever you're in Word, you would just click on create title page, and then you can include these APA style reports. The format's already on Word's templates. That one will work just fine. And it's important to include the title page and include stuff like page numbers in the bottom, everything like that. So in your introduction, you just have a title page, no abstract. And then your introduction itself is probably one of the most difficult sections. I have less of a problem with this because I write my introduction after I write everything else, because the background that's required for these is easier once you have already done your research and discussed your results, and then these are just important things to prompt and understand it. So they are three main sections in your introduction: background, the physiological question, and the goal and hypothesis. So your background is talking about how much of a good fit your your model organism. So in this case, it's this tropical field cricket. It's going to be called Gryllia signalis. It lives in South Florida, and it's typically used as a feeder cricket. And as you're going through here, you should talk about the equipment and metabolic, like the idea of calculating calories and how we're using a derived value. All of this information would be just summarizing how this experiment is being collected and why someone would want to look into this. Your second section is going to be the physiological question itself. The physiological question itself is going to be going over your actual hypotheses. So there are three main experiments in this lab report. You're going to have your control versus glucose at two different time periods, control versus stress, and control versus increased temperature. Those three experiments have their own key terms involved with it. So for instance, control versus glucose is talking about this idea of heat increase of feeding. So whenever you're looking up articles on that, it would be best to actually go into your previous papers and look into those. So if I go in there and open up the insect metabolism, I include this background information as a hidden slide in here. So this is specific dynamic action, and it's talking about how after feeding, it's going to have this sort of ideal peak of a metabolic rate, and then it's actually going to go back down the baseline. This is all based off of this respiratory exchange, where in order to actually utilize this energy that it's consuming, it needs to convert all of these sugars into glucagon, into proteins, and to utilize all of this new energy, it is converting it, and it is performing anabolic and catabolic reactions that actually increase the metabolic rate for a short period of time. So in this case, it's talking about around 10 minutes is at its maximum, and then over time it goes back down. So heat increase of feeding would be an important concept to go over. Another important concept to go over would be this relationship between temperature and metabolic rate. You can imagine, because this cricket is a polyotherm, it is ending up in most of this in this lower section, which its body temperature would actually be determined from the outside, and because it doesn't regulate its body temperature, it's a polyotherm. So it's actually adjusting its body. Its body temperature is dependent on the environment, and because of that, it's going to have a reduced metabolic rate at a lower temperature and an increased metabolic rate at a higher temperature, just because it can't actually regulate and stay at its normal body temperature across time. So it actually is dependent on its environment, on its activity. So that directly changes the amount of respiration that's possible. So that within this thermal neutral zone, it's going to, this is talking about homeotherms, but its metabolic rate is variable for whatever. This is more involved within homeotherms, but the polyothermy is the key physiological process that's being observed in this. So, and then stress, the relationship between stress and metabolic rate is related directly to stuff like flight or flight response and stuff like that. So addressing your physiological question is important. Goal and hypothesis is where you're going to reference the physiological questions that you're citing. So these ones would be a good idea to find citations for your physiological question and then address them in your goal and hypothesis. So your goal is what can be learned. The hypothesis is your actual prediction. So based off of this physiological activity, you're going to predict that something's going to happen, and you would write that out explicitly as a separate paragraph. And that's 2 points. So just make sure you write your goal and hypothesis at the bottom. Now, materials and methods, these are pretty simple. What it means by written and listed is talking about how you're going to write out your methods. So if you look at other papers, you'll see that whenever they're going through these, they're describing each of their individual experiments, how they filtered their populations and stuff like that. But they're going to talk about how they measured their results in different sections of the actual paper, depending on which experiment's going on. And then they're going to describe them in detail in paragraph form. What is required for us is, because we're working with 3 different experiments, is going to be talking about prep and then the actual 3 small paragraphs on how we actually performed it. It can be fairly short. These are only 4 points, and it is just more going over what you guys did. Just note that you should not write it in the second person. What it's talking about for that is, you shouldn't write it in the student did X, or the student did Y, or the scientist prepared the experiment. You're going to be talking about it in either first person or in third person passive. So third person passive is what you want to write all your lab reports in, but in the appendix for these, it can be written in first person due to the actual rubric itself, but it's important that this is just talking about what activities were done. This isn't the methods and how to actually perform the experiment. It's talking about what happened. And then the last thing for these is including a bulleted list of all your materials. That is a single point, but this list of equipment and materials, you would be writing out all of the equipment that was used in the experiment. It's included in the in the actual if you actually go to animal physiology and go into your animal metabolism lab manual, it's going to include all of that information. So first of all, it has a little bit of information on background that you can include. So a lot of your introductory background is included within this PDF, but for materials and methods, you're going to include this sort of bulleted list. And while it's asking not to copy directly from here, it is not a major thing for these, but stuff like Power Lab setup and everything like that, you can see that this is talking about it as an instruction manual. For our actual materials and methods, you would be just talking about what was happened to it. So the spirometer pod was zeroed. The equipment was set up by connecting the gas analyzer to the Power Lab equipment, and then the cricket was placed inside an environmental metabolic chamber to actually perform the experiments. So whenever you're writing these, you would be describing them and then talking about the individual 3 protocols. And you can see that in there they talk about how you have this 30-minute control and an hour of recording. These are all important information that you would need to include in your methods, but otherwise you wouldn't include a lot of you wouldn't include a lot of the recording instructions in here. You would just talk about how the data was recorded and included in the LabTutor software. Another important part of this is calculating metabolic rate. You would want to include any equations that is run. So this is obviously volume of oxygen consumed in 1 hour over the weight of an insect. So this is VO2, and you can also calculate the milliliters of CO2 milliliters of CO2 consumed. Milliliters of CO2 released as well. So, and whenever this is calculating their metabolic rate, they're actually looking at the metabolic rate of CO2 released whenever they're measuring these. But in ours, we're measuring oxygen consumed, and that would be included in your PowerPoint. If you know how respiration occurs, you can see that for 6 oxygen, 6 carbon dioxide, there's a certain ratio between them, but you should note that RER is actually, well, the respiratory quotient values have a certain ratio between those two values. So if it's eating lipids, for instance, if your cricket was eating lipids, the respiratory exchange ratio changes. And that means that there's going to be a deficit in the change in metabolic rate whenever you're measuring these, because this is not actually measuring the calories burnt. This is only measuring the CO2 released or the oxygen consumed in reactions across the insect's body. But you need to include that list of materials. Now, for graphs and results, you want to include at least 3 graphs or tables, so 3 figures. Because we're working with 3 different protocols, that's really easy. You would just separate it into 3 separate graphs, including your formatting. It should include a title. It should include your units and your and axes labels, and then you should include your your axis label, and you should include a title for, you've already included your title. You should include a figure description at the bottom. And in terms of graphing a bar plot and stuff like that, you want to include statistical analysis on there. So, for instance, for your bar plot is including error bars. For your histograms, if you're looking at a Box-and-Whisker, the actual Box-and-Whisker itself is a form of statistical analysis. And then for line plots like this, you want to include this R² value, and you don't really need to include the 7.99e, but you want to include this power value up here. Like, that's the important value for it. That's just an intercept. But just note, whenever you're calculating these graphs, that you should include each one of these for full points. So that's formatting. That's 4 points related to that. And then narrative of results. This is separate from your actual results. So if you look at a actual experiment, you can see, well, first of all, these are statistical tests, and that's how you would report it in the same way. You would include a table, and then you would include a description of your table and results for that. A better view of that would be this ANOVA, but for your results, you're actually going to have paragraph reports of the results separate from figure descriptions. So this figure description is just describing what this figure is, but it's not talking about what was observed in the experiment and how does it compare to other papers. But whenever you get into actual discussion, you can see that there's a lot more cross-references in there. So they're actually comparing previous papers in here, and then they're actually discussing the physiological process. But for your results, you're including, well, this is methods. Your results are actually talking about what observations happened separately and what statistical analysis were run. So that's where you would include your ANOVAs and everything like that. You don't have to include an entire ANOVA table, especially we're not performing two-way ANOVAs. We're only performing a one-way like this. So the key values on here, the degrees of freedom you wouldn't really need to report, but the p-value and the f-critical value would be important for running your ANOVA whenever you're actually running it in Excel. So for reporting your values, just make sure that you include paragraphs of what was observed at the time, or or a general overview of what was going on in that experiment. That needs to be separate from describing your figures. Yeah. And then your discussion of your results. Discussion is probably like this is going to be the bulk of your work right here, and it should be the most heavily cited section of your actual lab report. You can see in most papers they're describing, well, yeah, if you go into the discussion here, it's going to be talking about the physiological process and how that relates to your to the hypothesis. And then for each experiment they're talking about the individual results and how that relates. So like describing why these results happened rather than just reporting what they are. So for instance, in our discussion of our results, we would have 3 separate paragraphs for each section of your results. So for changes in, well, in metabolic rate for stress, you would have a couple paragraphs on there. Then you would have discussion of how that meshes with your hypothesis, how does it deviate from it, and stuff like that. And then you can describe what steps could be done to improve these results, or what other papers did differently from ours that changed their observed results. Cross references are not going to be a major effect for this experiment, but cross references will be more important for the crayfish experiment. So that's why I'm just describing other papers, just so you guys can know where I'm trying to get at whenever we're looking at these. And then agreement with the hypothesis is you can see that's less points in the actual discussion of results, because this is actually where your citation heavy. Agreement with the hypothesis is saying in each paragraph that you describe how this relates back to your introduction and your background. And then in conclusion, a summary of the paper is literally just a paragraph of what we observed and how that meshes up with your results. This would be very similar to what your abstract would be. And then your broader picture, this is actually talking about what happens outside of the lab. So for instance, because we're talking about metabolic rate and changes in temperature, how would that be important for a climate scientist or for a farmer that's trying to avoid plagues of crickets coming in, like the crickets that are currently taking over Denton right now, all of the brownfield crickets. So it's different species, but they're currently taking over Denton. It would be important for them to understand what happens if the change in climate increases the average temperature. Will that increase the actual length that these crickets are going to be around for, or what plants they can actually grow at that season? And then literature cited, you need to have a consistent format. So APA format is fine, but you should. It's very important for your literature cited that you have 3 external citations and the lab manual. So whenever you're going through here, you want to include, oh yeah, and then you also want to follow these additional points, but whenever you're citing these, you need to include literature cited for everything. You can also, like, it's talking about Google Scholar. I actually am producing a separate video on this, because actually observing these is very important for your literature cited, but you need to have 3 external references in addition to the lab manual within the introduction PowerPoints. I include an already formatted lab manual citation. It's a very long PowerPoint. Will it show up though? No, because it doesn't like me. Oh, yeah. Yeah, some more information on like what's it required for your graphs and tables. avoid plagiarism. Yeah, so this is your actual the actual format that I've already included for the lab manual. You would need to include 3 external citations in addition to that, and the best thing that you should include is a DOI link. This DOI link is the easiest way for me to be able to tell if your report is signed, well, if it's from a correct, well, if it's not from a journal of ill rapport. That would be the best way of saying it. So there's undergraduate journals, and there's certain citations that would not include these, like, for instance, a dictionary definition would not be an appropriate citation. You would want to cite an actual scientific article for your 3 external citations, or a chapter in a textbook. And those chapters in a textbook and everything, if they're available online, will almost always include this DOI link. And it is very important to include that, or to include a link to your actual article. Most of the periodical volume numbers and pages are antiquated because we don't buy the physical volumes anymore. We're looking at the DOI links. So this is the important information that you should include for all of your papers. But if it has a link online, if it's available online, you should include a DOI link. And then for ours, lab manual, including the lab manual is important. That's, I mean, yeah. And then you include page numbers at the bottom. Make sure that it's above 5 pages. Include all your units, and so this would be a deduction if you didn't include a certain equation that you were using throughout the experiment. And then grammar, I haven't marked off points on grammar. I can read most of your papers pretty easily. So we've gone over all of the requirements for the lab report. I'm going to get into how to report these values. Be sure to actually look over the appendix. Make sure that you're following all these examples. But I'm going to talk about how to report your MR data. So whenever you collect your data, it's available on Canvas. It's going to be called cricket metabolic data. This is going to include the metabolic rate of the cricket at the control temperature. It's going to include the metabolic rate after it's been fed glucose, and then after it's an hour after being fed glucose, it's going to have a slightly lower metabolic rate. Under the stress metabolic rate, this is talking about whenever the cricket's being exposed to sounds, being exposed to light, and it annoys the cricket, and it's going to enter a fight or flight response, and that's going to alter the metabolic rates. And then the increased temperature is another set of is going to be another set of temperatures and metabolic rates. So how are we going to calculate these? Let me first unhide this section. So how are we actually calculating these? You can see variance, means, and standard deviation. So means are the average value. This is going to be important for bar plots. You can see if I actually just delete all of these average values. First of all, it's going to be randomized each time, but whatever. If I delete all of these, how am I going to calculate it? I want to type in equal sign, and you can see I'm in this top area. I'm not typing it in here. I'm typing in the function bar. I type in equal sign AVG. Well, actually, it should be average. Yeah, so average, and then you would want to double click on that. You can see now it's saying average number 1, number 2. Then you would highlight and select this entire column. So from B to the B14, and then you'd press enter. And you can see that it's populated this value as an average of all of these values, but it's only calculated on this column. On this column. So what am I supposed to do? Well, you can see in the corner right here, there is this little green box. If I hover over it, it changes from a white plus to a black plus. If I drag that across for all these values, I immediately get the mean value for each one. So this is the average in this, well, for the center of your values. Now this is directly important for creating your bar plots. So if I just select all these values, I press the control button and select the titles, and I collect, and now you can see both of these are selected in gray. I can click on insert 2D column, and boom. But you can see that these are all the values reported together, and it's not including error bars. How do I add error bars? You do not want to just click on add error bars, because you can see that they're all the same height. That's wrong. You want to go into more options, and you want to click on custom error. And now the values for calculating custom error are going to be these standard deviations. Standard deviation and variance, you can see that variance is very high for some values, and then means are not very high for some values. So these variance values are talking about how widespread that normal distribution is. Standard deviation is reporting those same values, but it's not at the same extreme as variances. But both of these can be used for performing a T-test or an ANOVA. So standard deviation and variance are related with each other, but they're not the same values. And if you remember, I talked about that in the introduction. But means are these actual values themselves, so the bar plots. Whenever you want to calculate the standard deviation, you would type in STDEV, select the values in the same way, and then go across. This variance, we're not using it for these, so you wouldn't really need to calculate them, but you can see in here it's var. So let me get to these error bars. So you see I clicked on the error bars, and then because I already had this menu on the side, I can go into custom, specify values, positive error, you want to select, then negative error, you would highlight the same values, click on OK, and you can see immediately error bars are produced for each one. So you can see that for this increased temperature metabolic rate, this increased temperature metabolic rate has a very wide error bar on here, and that's because you can see the standard deviation is very high, and the variance is very wide, and so that's a good representation of these error values as you're going through. Now you can see that these say control temperature and these say increased temperature metabolic rate. You do not want to include two different types of values on here. So how you're supposed to only show control versus glucose or control versus stress. That's what this button comes in. It's a chart filter. So we've made one chart, but you can actually use it for all your other charts. So I can deselect all of these, click apply, and boom. This is nearly a chart fit for reporting. You just have to include your titles and your units on the side, and boom, you're done. So whenever you're calculating your charts, you should be able to first calculate your means, calculate standard deviation, and you should be able to output one of these, and this would be appropriate for all of your charts. So you can just create one, copy it into Word by literally just selecting it and doing Control-C, and then you can do temperature versus the increased temperature one. Click apply. You can see that there's an increase in temperature, but there's a lot more wider variance for this increased temperature metabolic rate. Or if you wanted to report the control versus experimental temperature, which is in degrees C, so this units on this side, it's going to change. If I click apply, you can then have that. So this bar plot is very flexible in what you want to produce. Now, for creating the Box-and-Whisker chart, you're actually going to be selecting all of these values. So if you select with the headers, so you can see that we're selecting the titles and everything. We drag that down, and we've selected everything. I can do insert, a histogram, box and whisker. I want to add a legend, and then now that the legend's in here, I can remove this section right here, and these are all labeled correctly. I can then remove the individual regions for each one, because you can see you can't filter on here. Yeah. So how we modify these is that you would highlight individual ones and remove them. And so this would be an appropriate graph for putting onto your Word document. Now, for plotting temperature versus metabolic rate, these values are a lot more difficult to start working with. So I'm going to go through this step by step, and this would be probably the most important part of this video, I would say. So please use this as a reference in the future, because creating these graphs and making them look nice, so you have different control and experimental, is a bit complicated. So I'm going to go through this step by step. These values are different than what you would see in your own experiment, because these are all randomized just by a little bit. So this is the kind of like the data that you're going to see whenever you receive it, and you can see temperature and metabolic rate, and then it has labels for control and experimental temperature. You want to highlight temperature and metabolic rate, and click on insert, a scatter plot, and you can see that you can see the trend right here for your scatter plot. And then you have this wide area on the other side. You would want to actually go into these axes options, not that width and height. I would want horizontal axis. I put it between 15 and 35, and that is a good representation of the data. But you can see these are the blue points without any labels on or anything. So you want to include a legend attached to it. I put the legend at the bottom. And as we add values to this, you're going to see that it's going to change the color of these. We also want to include a trend line on this, because you don't want to include it as a linear trend line though, because this relationship, if you see in this paper, so this is temperature report on the other side. So this is talking about moths. These moths actually are endothermic, so they are measuring the increase in temperature that the moths are voluntarily doing at different levels of oxygen. So in hypoxic conditions, there's very low oxygen. It's not able to produce, you can see in here, it's not able to produce enough body heat in order to start flying, which is required for getting their metabolic rate high enough in order to fly. And under control, it's able to reach those temperatures, but under hyperoxia, it performs around the same as control. So you can see whenever going through here, the VCO2, the metabolic rate consumption at different levels of oxygen, it's actually most comfortable in control, where under hyperoxia, it actually reduces its metabolic rate. So it's reduced performance under hyperoxia. So these are moths that have been conditioned to different levels of oxygen, and then this relationship is basically flipped from what we're seeing. So you can see that there's this exponential rate of change from a low temperature to a high temperature. And because they're measuring it based off of VCO2, it's the CO2 released that they're measuring. So as they increase the amount of energy that they're burning, their thorax temperature increases. What we're looking at is the change in temperature of a polyotherm. So as the change in temperature occurs, its metabolic rate is going to steadily increase. So in that case, we would be using either a power experiment or an exponential. Power, we wouldn't actually want to use, but you want to include the r-squared value and the equation on the chart. Now the reason why is because these values right here are important statistical markers. So the r-squared value is on a range between 0 to 1. So you can imagine if the r-squared is 1, all of these points would be on this line and show a perfect relationship. An r-squared value of 0 means that all of the points are randomly spaced out and have no correlation to the trend line. This y, this is a intercept value, so it ends up as 8 whenever it reaches the end. And the important value for an exponential graph is actually this value up here. So for every change in x, this is directly related with Q10 values that we would be looking at later on, because under Q10, every degrees, for every 10 degrees increase in temperature, there's going to be around 3 times an increase in metabolic rate. Well, it's going to be between 2 and 3. And that is very similar for even enzymes. So that's why we're calculating it as an exponential relationship, because these exponential relationships are directly related with Q10 values. Now, for these values right here, now that we have this trend line, it's kind of ugly right now. So we can extend the forecast, so we can go not 1 but probably 10, 5. I'm going to do 5 on each side. Yeah, this forecast is just extending the wings of it, so it's just showing how it's going to increase as it reaches 40 degrees. And these crickets would survive at 40, but this is the change in metabolic rate over temperature. This is the forecasted line. Now, how do we get those pretty colors? You want to go back into this chart filter, and then you want to select data. So we have the overall metabolic rate that we use for a trend line, but we do not have the control temperature or the exponential temperature as separate ones. So as a legend entries, you would include the name, the x values, which would be the temperature from 2 to 12, and then for the y-axis values, it's also from 2 to 12. And then you would click on OK. So now your control temperature, you can see, is labeled in orange, and then you can add another one for the experimental temperature. And once I perform that, you can see now we have nicely spaced dots where you have these values. You would also want to remove the label from here, and boom, we have a nice graph that you would also want to include a title for. You can report these values in like the exponential value and your r-squared value in your results narrative if you want to, but producing these graphs would be very important for the next experiment, because Q10 values can be represented this way, or it can be represented as the change in means. So how do we perform an ANOVA and a T-test? That is the final section of this, and again, it's fairly simple. So how to perform your ANOVA. So you see these values right here. We see a group of values right here. See your group of values for glucose, for stress, and increased temperature. Each one of these has their own normal distribution. That's what these variance values are talking about. So if I look at this as insert a histogram, well, I'll insert it as a Box-and-Whisker. If you remember the shape of this bell curve, it's going to be going, if you can imagine it being flipped on its side, this is the mean value right here. These are the edges of the variance, and then this is the edges of the actual data set size. So this is that big hump normal distribution, if you can imagine that. And this is just represented on the side. Whenever you have each one of these, you have them as individual sections. What ANOVA is doing is for groups of greater than 2, it's going to be looking at the sources of variation. So between groups is talking about how wide this normal distribution is between everything. So that's the p-value, okay, yeah. So the source of variation between all the groups is talking about how wide the data is between each parts of this data set. And then within groups is talking about the source of variation for each individual set itself. So we have 8 values in here. We have 14 values in here with a certain degree of freedom and a certain degree of variation, where within this group it's going to have its own different group, and each one of those has its own normal distribution. If the normal distribution within each group is too wide, it's going to increase that p-value, and that means decreasing the significance. So this is the actual statistical test at the end, and what you'd want to report is that your p-value is lower than 0.02, and for instance, 0.05 for this course. Yeah, so 5%, and that means that the overlapping of multiple normal distributions are outside of the range. So that means this edge of the variance is far enough away from these ones to consider that this is a distinct value from these ones. So it's a test. The hypothesis is that all these values are actually the same normal distribution, but as we can see, they are certainly not visually. That's why these graphs work well for visually showing the difference of variations. And then within each one, it's more similar within each point in here. So a point in control is more likely to be a point in control than it is within glucose, or glucose after 5 minutes or 30 minutes. That's what it's looking at. So it's comparing the source of variation between groups and within groups, and if they're significantly different, it's going to have a p-value of below 0.05. That means it is significant. And then your t-test, and how to perform your ANOVAs. You do not want to select your entire data set. You just want to select which values you're actually looking at in your experiment. So control versus glucose, so this is 3 different values, so you can't perform a t-test on it. But you want to go into, and now you can see add-ins. You want to enable this analysis tool pack, click OK, and whenever you go into data, you can do data analysis, and then you can actually perform your ANOVA. So we're doing ANOVA single factor. So your input range is these values right here, and then labels in the first row, and then I'm going to put it into a new worksheet. That's going to call it ANOVA, and you can see that this is the same single factor ANOVA that we saw before. The p-values are significantly different from each other. So that's what we're looking at. And then for your t-tests, you can see that it would be performed the same way, but you would only want to include, say, control versus stress, which is not a significant difference, I believe. And then if we look at that on here, yeah, control versus stress, you can see that the standard deviations are actually overlapping each other. So let's do another data analysis. That would be a t-test, assuming unequal variances, because we can see that the variances are 400 and 300. And so t-tests are actually dependent on the number of points. So this one is actually going to be wider. But variable 1 range, you want to include all of these. Variable 2 range, all of those. They have labels. Your alpha is 0.05. The hypothesized mean differences, this is talking about your null hypothesis. That's going to be 0. You're assuming that's going to be 0. And I'm just going to call this worksheet T. Oh, input range 2 again. Stress versus control. Yeah. So you can see on here that your t-tests are actually, well, they're just nearly significant. Yeah. So this is your one tail and two tail t-test, and these would be the values that you would report in here. That's it, I would say, for these sections. Be sure to look at these, so be sure to consider how you would write out your lab report, where you're going to start going broad, going into the specific aims, and then go into the actual presenting the key results and findings, and then discussing what they mean and bringing it to the outside world. Talk about the limitations of what you've performed, and then finally, whenever you're exiting your discussion, you're talking about how this can be applied to the outside world. And